Okay. So the first thing I should say is, well, thanks for inviting me to give this presentation. It's getting kind of boring sitting around the house, so it's nice to get out. Um, you'll notice that I'm a professor of physical oceanography, not biological oceanography. So you might wonder why somebody like me would be talking about red tide. The bottom line is that ecology is not biology, it's the whole shebang. And as you'll see, the circulation of the West Florida shelf is extremely important in determining, first of all, whether we will get a bad red tide in any given year, and then where that red tide will actually manifest itself as a nuisance. So let's see, I guess let me figure out how to do this. Which button do I hit here to?
Hey, Bob, I think you just let Nicole know that you want to advance the slide and she'll advance the slide.
Okay, all right, good, great. All right, so here's the order of my presentation. There is an ongoing program in the College of Marine Science, University of South Florida. It's called COMPS, and it gets support from NOAA IUs through SICORA. And we also have some support from the National Academy. And so I'll kind of give you an introduction to what we're doing, the observations and models that I believe are germane to your program. And then I'll make application to the 2018 red tide bloom. And when I'm done with that, I'll give some other application examples that might be of interest and then entertain questions. So next slide, please. Okay, so the COMPS program, Coastal Ocean Monitoring and Prediction System, it was initiated in 1997 from the Florida legislature, and there are a number of ecological questions that were germane, and so we were able to get a little bit of support from the state to initiate this program. The left-hand map of the West Florida shelf in the state of Florida shows a number of buoys that are presently deployed, some of which are giving real-time data available on the website that you see there. And the right-hand panel is a large-scale model. We call that our West Florida Coastal Ocean Model. And it's designed to downscale from the deep ocean across the entire continental shelf and into the major estuaries, including the Charlotte Harbor Sanibel Estuary Complex. We get our open boundary values from the Naval Research Lab's Gulf of Mexico HICOM. So it also includes the forcing of the continental shelf by the deeper Gulf of Mexico. And then using an unstructured grid model, we go from the resolution of HICOM at the open boundary to about 100, I forgot if it's 100 meter or 150 meter resolution going into the Charlotte Harbor Estuary. So that's kind of the thing that we're doing. It's a coordination between coastal ocean observations and coastal ocean models to try to understand what controls the water properties on the West Florida continental shelf, and therefore what controls ecological issues like red tide, or also fisheries recruitment, or any subject of societal importance that has its origins with the coastal ocean. So let me move on to the next slide, please. So this West Florida Coastal Ocean model, as I said, it downscales. So you're looking at three successive panels from the left to the right. The first one kind of is a blow-up of the Tampa Bay to Charlotte Harbor area. This is the same model. I'm just zooming in a little bit. And then the middle panel zooms in further to include the entire Charlotte Harbor estuary complex. And then the far right panel is a further zoom at the model resolution to look at the region near the inlet. So as you can see, we include all of the inlets. It has 8 tidal constituents And what you're looking at is velocity superimposed on salinity at a time of I guess, outgoing tide. And these model simulations, they're automated, they're done daily. They're available at the website that you see posted there. And what I'm showing for today was forecast in the early morning of 2 days ago when I made this PowerPoint presentation. So we provide a nowcast and then a 2-day forecast. It's actually a 3.5-day forecast, but what we post on the web is a 2-day forecast. So anybody can access this any day, and it's value to recreational and commercial boaters. And it also has application to any ecological problem that you may be considering inside the estuary. And, you know, I listened to the last talk and I saw that Dr. Chen is getting his open boundary values from RWFCOM. And so it's nice to hear that, because frankly, you can't really consider what's going on in the estuary without considering what's going on in the adjacent coastal ocean. Other applications of this, well, I'll talk about that later, but let's have the next slide, please. All right, so let's get into this 2018 red tide bloom. There are certain things we know, certain things we don't know, and certain things we can or should be doing about it. So the knowns are that Florida red tide is a bloom of a toxic dinoflagellate called Karenia brevis, and it's quite naturally occurring. We can go back in the literature and see that we've had major red tides in this region going back to the 19th century. And if you go back historically, red tide is actually alluded to in the logbook of the fellow that chronicled DeSoto's voyage into the Gulf of Mexico. So we've had this for a long time. It's nothing new. It didn't start with the sugar industry. It didn't start with Florida agriculture. It's been with us for as long as we can determine. It initiates offshore under oligotrophic, which is nutrient deplete conditions, which is a little non-intuitive because we keep thinking that nutrients are the culprit. It generally manifests as a nuisance once it's transported to the coastline. And so both the offshore conditions and the transport are determined by the ocean circulation. That's why the ocean physics are so important to this phenomena that we call red tide. What don't we know? Well, do humans exacerbate red tide blooms? And the answer is probably yes, but we don't know exactly how yet. We don't know what terminates a bloom. And we don't know if there is a viable means for mitigating a bloom. So what do we do about it? Well, first thing we need to do is have more observations. The scientific method begins with observations. We look at something, we question why it is, we make hypotheses, we test those hypotheses, and maybe we learn something. And so we need to support full water column observations across the entire West Florida continental shelf. You can't just focus in on where the problem manifests as a nuisance, which is pretty much what we've been doing up until a few years ago. You've got to look at the entire system as a system, see how this process evolves in order to try to understand how it works. We need to support necessary modeling and tracking, and we need to conduct laboratory and microcosm mitigation experiments. So, years ago, there was an attempt at using clay to flocculate the red tide organisms, send it to the bottom and kill it. There were also other experiments using toxic materials to try to kill the red tide. It's very dangerous to do anything like that. So before we do anything like that, we really need to pay attention in the laboratory under controlled conditions. And once we feel comfortable with that, go out and maybe do some microcosm or smaller mitigation experiments in localized regions. You know, we're hearing the news these days about some experiment performed in Wuhan, which got out of the laboratory, and now that's why we're all cooped up in our houses. So we don't want to do anything about red tide until we really know what we're doing. And we want to solve known human-made problems. There's been a lot of talk about Lake Okeechobee. There are problems associated with Lake Okeechobee. These problems can be solved. They are human-made, and we ought to deal with those once and for all. Next slide, please. Okay, so let's see what's going on the West Florida shelf. On the left-hand panel, you see that map again, and there's an arrow pointing in the lower left-hand corner of the shelf that we call the pressure point. And that's a very critical location in determining water properties on the shelf. I can kind of go back and say that William Shakespeare knew all about this problem in the 16th century when he said, to be oligotrophic or not to be oligotrophic, that is the question. So it really boils down to, how does the shelf get its nutrients? And so the shelf is very wide, and we know that nutrients coming off the land get consumed by phytoplankton very close to the land, within about, say, 10 kilometers. And a good measure of that is where you see the salinity fronts that are coming off of the estuaries and off the land drainage. We also know that the continental shelf gets nutrients from the deep ocean. And under most conditions, the movement of those nutrients onto the continental shelf is also limited to a very narrow band near the deep ocean, near the shelf break. And that boils down to about 30 kilometers. So with the input of nutrients from the deep ocean being confined and the input of nutrients from the land being confined, that leaves a very broad middle portion of the West Florida shelf, which is generally nutrient deplete, and why we call that oligotrophic. It is under those oligotrophic conditions that Karenia brevis can outcompete other phytoplankton and therefore become a bloom. And once it blooms and takes over from the other phytoplankton and gets carried into the shoreline, then we suffer, a red tide bloom very close to our own hearts. The right-hand panel shows all of the red tide observations collected by the Florida Wildlife Commission from 1953 through 1915. And what you see is a very dense concentration of those observations between the Tampa Bay and Charlotte Harbor estuaries, a little bit north and south of that as well. And then you see kind of a blank region in the Big Bend, a blank region in Florida Bay, pops up again a little bit in the Panhandle, and a little bit on the East Coast. But the real epicenter of where we suffer red tide blooms is Tampa Bay to Charlotte Harbor. And you know what? It has very little to do with Tampa Bay or Charlotte Harbor, as you'll see. Going back to the left-hand panel again, where that arrow points to the pressure point, when the Gulf of Mexico loop current hits the West Florida shelf slope at that location, it can actually set the entire West Florida shelf in what we call an upwelling circulation, which means that water will be flowing toward the shoreline along the bottom and flowing away from the shoreline at the surface. So whatever is cooking offshore at depth can be carried to the shoreline when the loop current is doing that, and whatever is at the shoreline at that time can be carried offshore. So that's a very important thing to remember. And in years when the loop current is doing that, we can change the whole structure of water properties on the West Florida Shelf. We can take what had been an oligotrophic region and make it a nutrient replete region. So if anybody ever wonders, how is it possible for a shelf that's defined as oligotrophic, how is it possible for that shelf to support such abundant fisheries and have these copious algal blooms? Well, it's because it's not always oligotrophic, and it's the ocean circulation under very special conditions that determine the water properties and consequently what can grow in that water. So can I have the next slide, please? So this is what happened in 2018. The bottom panel is a log scale of Carenia brevis cell counts from June 1917 through November 1918. A canonical red tide season is one in which the bloom will appear sometime in fall, late September through November, and last for a couple of months and go away. And we suffer for a little bit, but not too badly. In October of 17, a bloom arose that year, and it didn't go away. It's like the unwanted relative that comes to visit and stays. And so we had this 17 bloom that made its way well into 2018. And then in September of 2018, that bloom got augmented by a new 2018 edition. What the colors in that plot indicate, the red indicates when we started seeing red tide at the panhandle, and the blue indicates when we started seeing red tide on the East Coast. And the right-hand panel shows the distribution of red tide concentrations averaged over the whole month of September 2018, and you can see in that plot that we had simultaneous observations of red tide blooms both on the East Coast in what I call the epicenter region and as well as in the panhandle. And I contend that those three locations where we had red tide bloom all originated in what we call the formative region, which is a little bit to the northwest of the Tampa Bay region. Okay, so 2018 may have rivaled 2005 as the worst of the recent red tide bloom years. And as I said, those two factors were in play. For some reason, it never went away in 17. And then we added on top of that a new bloom in 2018. And it's been questioned whether or not Lake Okeechobee, the releases of water from Lake Okeechobee, exacerbated the existing bloom. And I'll say this is a very good question, but to date, we really lack a very definitive answer to that question. Next slide, please. So what did USF do in terms of field work when the 2018 bloom popped up? We went ahead and deployed what we call a glider. It's a robotic sampling device that we could send offshore, and it operates autonomously. It samples water properties, sends these back whenever it surfaces, so several times a day. And so we're able to go out and track water properties. The right-hand panel shows the track line. We started in that north location on August 24th, and we ended on September 17th. There's a little loop that you see around September 4th. That's when Tropical Storm Gordon went through caused the circulation to go northward, and it was a little too strong for the glider to contend with, so it went through a little bit of a loop until Gordon went by. The currents switched during Gordon from being upwelling favorable, because the loop current at that time was impacting the pressure point, to being northward for several days and pretty strong. And so when we ran our models, we actually determined that red tide cells that may have been to the north of where you're looking right now, the north of that glider track, those cells could have been transported to the panhandle. And sure enough, it was shortly after Gordon that we saw red tide cells start to light up in the panhandle. Next slide, please. So this is what the glider observed. You're looking at from the top left panel is temperature, the bottom left panel is salinity, top right is chlorophyll, and the bottom right is oxygen. And what you see is relatively cold water at depth and warm water on top. The cold water is salty, so it actually originated from offshore. It came onto the continental shelf from the deeper ocean. And if you look at the chlorophyll, what you see is that between about the 30 and 40 meter isobaths, the chlorophyll kind of lights up with high values at depth. And we see those relatively high chlorophyll values everywhere that the glider sampled, but only between depths of, say, 25 meters and 40 meters. So it wasn't in the deepest water, and it wasn't in the shallowest water. It was right in what we think is the formative region for a red tide bloom. And where we see the highest chlorophyll, we see low oxygen, indicating that there is some decomposition going on. And so the red tide was killing fish, even out there. Next slide, please. So we took our West Florida coastal ocean model, and every place where the glider had been on that particular day, we initialized that model with particles. Just inserted a particle in the model, and then we tracked, using the model velocity field, we tracked where that particle would end up over the next, I guess, 45 days. And actually, I misspoke. It's only actually just for the next several days. So it turned out that by October 1st, you can see that just about all of those particles made it to the shoreline, and you can see where the particles landed, from just north of Tampa Bay to just south of Charlotte Harbor. And so the epicenter region of red tide that we've observed from 1953 through the present time is consistent with offshore generation and advection to the beach along the bottom. That's where the red tide would end up. So, and this isn't the first time we've done experiments like this. We've demonstrated this before. So it's becoming fairly well recognized now that in most years, red tide originates offshore in what are generally oligotrophic waters, and those red tide organisms then can be advected to the shoreline. It's a real nasty critter because it kills fish, and those fish decay. The fish in their decay leave nutrients in the water, and the red tide then gets its nutrient supply from the dead fish. So once a bloom is initiated and can start killing fish, it doesn't need any help from us. It actually takes care of itself. Now, if we dump additional nutrients into the coastal zone, especially of the organic variety, which the red tide organism really likes, yeah, that will exacerbate the bloom. So we probably do, as humans, exacerbate these blooms, but we're not necessary for the bloom to actually form. Okay, next slide, please. Yeah, I should have mentioned, we also did those particle trajectory experiments, and we also demonstrated that from that same region we can get red tide to the Panhandle and also get red tide around the Dry Tortugas into the Gulf Stream and then up the East Coast. So that's fairly well established as well. All right, so once we have a red tide bloom, what do? Well, we do some short-term tracking using our West Florida coastal ocean model and also using another model that we call our Tampa Bay coastal ocean model. So if you look at the left-hand panels, you see a map of the entire domain, and you see little boxes. You can click on any of those boxes to zoom in on a particular region. So just to the right of the bigger box, there's a Tampa Bay box and a Charlotte Harbor box. And you can see then where the red tide would go. We do the same thing at much higher resolution for Tampa Bay. Our Tampa Bay coastal ocean model actually goes down to a resolution of 20 meters. And so we do the same kind of tracking in Tampa Bay. And then, since these plots may be a little difficult for the average person to look at, every Wednesday and Friday we come up with a user-friendly map, color-coded, to basically indicate where, or on what beaches and what regions we expect the red tide to be the worst over the next 3.5 days. The data that we use come from the Florida Wildlife Commission, from their lab in St. Petersburg, and they pass those cell count information to us every day, and we then do this tracking every day. We also try to do a seasonal prediction. Will this year be a bad red tide year or not? And so it's a qualitative thing. And we base those predictions on what the loop current is doing during the spring and early summer months. So remember, we tend to get our major red tides starting in fall, late September, October, November. And what's going to happen then along the shoreline in fall, we believe, is determined what's happening in spring and early summer in the formative region. So around June, I usually come out with what I think will be the seasonal forecast. Will we or will we not experience a major red tide? That forecast is not infallible. I always like to joke that the University of Colorado people, they come out with a hurricane forecast every year around this time of year, and their forecast gets better and better and better until the last year of the hurricane season, where they're spot on. And so, it's very difficult, as Yogi Berra said, to predict something in the future. But the scheme that we put together seems to have worked in 20 out of 25 years for which we had concurrent red tide and ocean circulation information that would allow us to say whether or not the loop current was impacting the West Florida Shelf slope at that position that we call the pressure point. And so I'll be making some kind of prognostication later on this year. It's too early right now to do that, but I will be doing something shortly. Now, having said that, we were doing this, I think we started doing this in 2012 or 13, but I was doing it quietly. I'd knock on my neighbor's door and say, hey, I think we're going to have a red tide this year or not. and I didn't go public. And then 2016 came along. I got a little cocky because I was correct in 13, 14, and 15. So I said, well, why not do it in 16? So I went public. And sure enough, it failed miserably. I said we were not going to have a red tide, and we did. And that red tide lit up in mid-September, about a week or two after there was a major sewage spill in Tampa Bay. So 2016 may have been a year when human beings actually contributed substantially to the occurrence of a red tide. That's speculation. We don't have any real hard evidence of that, except I didn't think there'd be one that year, and there was. So while we have a canonical description of what red tide does and how it evolves, there are surprises, and I'm sure those surprises will continue. It's a very complicated problem. Okay, so next slide, please. So what is the pathway forward? Predictions require understanding, observations, and models. And K. brevis, like anything of an ecological nature, is an interdisciplinary problem. Improved K. brevis capabilities will not be achieved by studying the organism biology alone. I mean, I say that emphatically. Please go back. Yeah, okay. And similar can be said for fisheries management. And at least in the red tide world, we are beginning to understand and accept this interdisciplinary approach to a very complicated problem. In fisheries management, I haven't seen that openness begin yet, and I think we need to be doing that. That's an editorial comment, an op-ed piece, so to speak. All right, observations. Well, we've got to have these regular glider surveys, and they have to be across the entire continental shelf, especially across what we think is the incipient bloom region. Gliders can't collect water, so gliders can't actually do the cell counts and the nutrient determination. So when we see something that looks as if there might be a red tide brewing, we've got to send ships out and actually collect the water and do the cell counts. And we've got to have strategically placed buoys, and we should start outfitting these with near-bottom nutrient sensors. Nutrient sensors are finally available. How robust they're going to be for time series observations remains to be determined, but we can at least begin that. We actually have buoys in the right place, and these buoys should be outfitted with nutrient sensors near the bottom. We need to make sure these models are funded, and we need to make sure that they can be used because they've been very effective. Pinellas County used our 3.5 day forecasts to position their response equipment in 2018. They were very effective at getting the dead fish out of the water before they came to the beach. And so that in itself was a great mitigation factor during the 2018 red tide bloom. And I have to comment that ecological prediction is very hard. It only takes 7 variables to predict the ocean circulation. It takes a lot more than that to start predicting anything of an ecological nature. And the unknown parameterizations, the so-called tunable parameters that you may hear about, they're a lot more complicated, a lot less known, and they vary over much larger extents than anything used in circulation. So I don't think that we can just implement ecological models for prediction. I think that's greatly overrated. And to actually do better ecological predictions, we need to apply an Occam's razor approach, kind of like we did with the circulation. I mean, boil the ecological variables down to what are the minimum number that we need to comment on K. brevis red tide. And being that K. brevis red tide is a very complex organism, that in itself is going to be a very difficult task. But, I guess the sooner we work on it, the better off we'll be. We still don't know what determines K. brevis bloom termination, and that may hold the key to mitigation. So there's still a lot more to be done, and I apologize for my dog, but he's starting to bark at somebody. All right, next slide, please. All right, so that's the end of the red tide, well, more or less, but let's just look at a couple of more applications. So what we're looking at here are an August realization of red tide trajectories. You had serious red tide in August of 18, and a November 19 one. You also had serious problem in November of 19. Just to the right of those panels is the salinity and velocity in the Charlotte Harbor estuary complex. So in 18, you had an awful lot of fresh water in the entire bay. In 19, not so much. And so, you know, the point is we just can't blame red tide on regulatory releases of water from Lake Okeechobee. Sometimes you get a red tide when there's a lot of fresh water in the system. Sometimes you don't. And that's not to say the lake isn't problematic. It is, but we simply can't blame red tide on it. Next slide, please. So there's been some talk recently about offshore aquaculture. In fact, there's a firm that has put in a permit to implement an aquaculture system offshore Sarasota. And the question I addressed is, you know, where might stuff that comes out of these fish bags, where might it end up? And so you see a simulation using our WFCOM showing that under upwelling conditions that are not uncommon, it may simply be a matter of days before what may come out of the fish bag could end up on Sanibel and Captiva beaches. So, you know, it's important that we understand how the West Florida Shelf works in order to understand what might impact people in the Charlotte Harbor region, or in any region for that matter. Next slide, please. So what are some other potential applications? Hurricane storm surge simulations and inundation by lesser storms as sea level rises. We've actually made some nice publications on hurricane storm surge impacts on your region. We explained years ago why Charlie broke an inlet in North Captiva Island. And most recently, we looked at the Irma responses of both the Charlotte Harbor estuary and the Tampa Bay estuary as Irma went by. And these are all published and available if you're interested. Informing search and rescue operations and tracking harmful substance spills, oil spills, and doing forensic studies pertaining to the origins of objects floating onto the beach. I did a recent pro bono work for a detective that called me up from Longboat Key. A body, unfortunately, a dead body floated up right near New Pass, and they had the timing of when this person was last sighted at a marina that was just inside the intercoastal waterway near the Cortez Bridge. There's an inlet just south of the Cortez Bridge, and the detective asked, is it possible that this person could have fallen in the water, been washed out through the inlet, and eventually down to New Pass? And so we did a simulation given the data that he sent us. Recall that our Tampa Bay coastal ocean model with 20-meter resolution can actually resolve all these features. And sure enough, we were able to lend plausibility to this guy's hypothesis. And so apparently there was no foul play involved. There's just a terrible accident. We've also been involved in some other forensic studies where there were murder involved, and we're able to assist in those cases. We've subsequently built a tool so that if somebody calls us up, gives us a time and a location within probably within minutes of receiving that information, we can provide a forecast of where either that harmful spill or that person will go. So we've tried to put together an effective tool to assist in any of these issues. As I said, we have 20-meter resolution for Tampa Bay. It includes Tampa Bay, Sarasota Bay, the Intracoastal Waterway, and all of the inlets connecting these with the Gulf of Mexico. We could do the same thing for Charlotte Harbor. I see that Dr. Chen has upgraded the model that he uses. I assume it has flooding and drying. I assume it's fully three-dimensional, but I don't know. But we're available if that could be of use for any of your activities as well. So with that, let me thank you for your attention, and I'll try to answer any questions that you may have.
Thank you so much, Dr. Weisburg. Would you mind putting yourself on mute for a second?
Okay.
A fantastic study, and it sounds like it's only going to get better with more observations. So lots to look forward to. Hey, we're close on our agenda time, but we have a lot of questions queued up. So I'd like to get through a couple of those. I think these are really good questions. And if you could do your best just to get through them quick, that'd be good. We have a little bit of we have a little wiggle room with our agenda time, but we don't want to bleed too much into it. Let me ask the first quick question, Bob. Let's say there was a 2,000-ton reduction of total nitrogen coming out of Lake Okeechobee, and that was done through different observations. Is that something that your COM model could handle in terms of the ecological response with chlorophyll?
Okay, so right now, as of today, we do not have the biological module of this model operating. We could do that in the future, and that is in the plan. Right now, we're just doing circulation. To answer your question, though, it would probably be help. It will probably be of help locally, because I think human input does exacerbate or can exacerbate an existing red tide. But I don't think it would matter with regard to the generation of a major red tide. We're still going to get those. It may last less long. It may be of less intensity. But the fact that the critter originates offshore and the fact that it gets carried to the beach, killing fish and therefore making its own nutrients, what you suggest would have an impact, but it's not going to be it's not going to mitigate the red tide.
Okay, thank you very much. So let's start at the top here. So first question: does this model include forecasted incoming tides? And I think the question is about lunar tides.
So the answer is yes. We have 8 tidal constituents in the model. We've tested the model for tides. It is fairly accurate. And so I would say that the tidal forcing for the Charlotte Harbor estuary complex is pretty good.
Great. Next question. Are there data along the eastern wall of Charlotte Harbor that suggest advection up the east wall from further south? For example, the Caloosahatchee River via Pine Island Sound.
That's a great question. The answer is yes, but it takes it takes some fairly nice wind forcing to do that. Under kind of calm conditions, you can think of those two regions as being fairly separate because the water depth is is fairly shallow in there. But if you get strong southerly winds, what's coming out of the Caloosahatchee will be sent north, and under strong northerly winds, the opposite will occur. So yeah, the two branches of the estuary system do communicate, but it would take some nice winds to do that.
Got it. Thank you. Another question here related to climate change. Are you seeing any changes of this portion of the loop current? I'm assuming that's the pressure point due to changing ocean temperatures.
As of right now, I would say that climate change has not really impacted, or anthropogenic climate change has not impacted the loop current in the least. And but there are large multi-decadal variations that occur naturally that do have impact on the loop current. And so, for example, the Atlantic multi-decadal oscillation that you may have heard about impacts both rainfall for the state of Florida as well as the strength of what we call the overturning circulation, which the loop current and Gulf Stream are part of. So the natural variability is actually having a much larger impact than the anthropogenic climate change aspects, at least up until now.
Great, thank you. Here's a biology question for you. Has research in harmful algal bloom allelopathy provided any potential solutions for mitigating red tide blooms? I've been advised that types of seaweed may show promise in disrupting the life cycle of Corynea brevis. Have you found this to be true?
So I really can't answer that question other than to say that that would be a viable experiment to perform both in the laboratory, which it probably has been already performed, as well as in microcosm. But until we do that, it's going to be difficult. But I have to comment further that if red tide had a limited spatial extent, it would be simple to mitigate. But it's spread over such a huge area, and the advection of new cells in occurs over a sufficiently long period of time that even if you were to wipe out the red tide in a reasonable size area, it would come back. And so the mitigation problem is really a difficult one. There's no simple answer to it, but I certainly am in support of doing laboratory and microcosm experiments.
Yes, it is. Okay, another question here. It's a correlation. Is red tide occurrence and severity correlated to hurricanes and tropical storms?
There have been some suggestions of that. I really don't think that they are. It's coincident that the peak of the hurricane season coincides with when we typically start seeing a red tide. And what a hurricane does do is it dumps a lot of fresh water on the land. The fresh water flows off, of course, and it makes fronts in the coastal ocean. Fronts are means of aggregating material. So let's say we have red tide cells coming in, totally irrespective of hurricanes, but now you add another aggregation mechanism by intensifying the front that will aggregate the red tide. So I think it has more of an indirect effect than a direct effect.
Great, thank you. So this question, let's see. So it's about, if you can recall your thinking in June of 2018, when we were already in a bloom and we didn't know, but it was about to get much worse. What was your prediction?
So it turned out that 2018 was a year when I predicted there would be in addition to what was already going on. So I predicted that 2018 was going to be a terrible year. And I think I did go public on that. What happened in September, or starting in late, I forgot it was late August or early September, after the bloom had already been in place, the loop current at that point sidled up against the pressure point and initiated an upwelling circulation that then persisted for quite some time. So whatever was offshore, it ensured that whatever was cooking offshore would come into the beach. And that's why I knew when we sent that glider out and actually saw it, I knew what the answer to the simulation was going to be. And I knew why we were going to get, such a terrible red tide, especially adding on to what had existed from, if I remember correctly, prior to September, the red tide was limited from Venice Inlet south, and September came along and all of a sudden it lit up from Venice Inlet to northern Pinellas County, and that was due to the new cells that were advected in by that loop current impact with the pressure point.
Great, thank you. And I think this is the last question that I've collected, but although there might be some more, and we're just a few minutes over time. Would you please revisit the connection with red tide and fish farming, offshore fish farming, cage farming in the Sarasota area? Please.
That's another great question. So there's two issues at hand. Number one, regardless of red tide, would stuff, whatever that stuff might be, could that stuff come to the Sanibel beaches? The answer is yes. Now let's get to red tide. You put a fish farm out there, you're feeding it all kinds of food, which is a nutrient source. And of course you have the fish themselves, which are putting organics into the water. And it is in a location that is otherwise oligotrophic. So that fish farm itself could become a factor in facilitating red tide bloom.
Fantastic, thank you. Are there any more questions for Dr. Weisburg that haven't been typed in?
Hey, this is Mark Schramek, National Marine Fisheries Service. Can you hear me?
You're loud and clear, Mark.
Roger that. Great, thank you. Dr. Weisburg, great presentation. Thank you so much. This information is very valuable. Just curious to the CHNEP, will this presentation be available for download in the future?
I don't have any issue with that. You're welcome to have it.
All right, very good. Just useful information because I do work on the Lake Okeechobee schedule release working group, if you will, and information in this might be helpful to direct the Corps and future discharges for Lake, and certainly in the two estuaries. But thank you so much.
And just to add on to that, when we do our short-term red tide tracking, we actually retain all of those. And so I forgot what year it goes back to, but we can go back, at least a half a dozen years, maybe more, that we've been doing this. And at the same time, we've retained the maps of salinity and currents in the Charlotte Harbor region as well. And so if it's of use to look at the coincidence between the appearance of red tide and the regulatory releases of water, that can be looked at as well.
Thank you. That might be very helpful for making, previous informed and possible future predictions with the red tides and Lake O discharges as well. Thank you, Dr. Weisburg.
Okay, and you're welcome to, contact me and we can discuss it.
Hi, this is Chen Chen. Dr. Weisberg, I just want to thank you for providing us with the boundary condition for the main flow for Peace, Low Peace River, and Shell Creek. Thanks.
Yeah, very good. I'm glad. I remember you started that with several years ago, and I'm glad we were able to be of assistance.
Yeah, it was like at least 5-6 years ago. Yeah.
Right. And as I said, we do have. I think the resolution of our model is probably higher than what you presently have, and it is available on a daily basis. What we have not included yet in the model, but which is very important for salinity, is to put evaporation minus precipitation in there, because the evaporation actually does significantly raise the salinity. So we've noticed in doing our data comparisons that we are underestimating salinity. So having evaporation is important. But all of this, all these additions, require support, and we're doing what we can do given what we're getting. So happy to interact and work with you guys.
Good. Thanks.
Great. Thank you so much, Dr. Weisburg. And just the final plug that I feel obligated to doing it because it paid for my PhD education at the College of Marine Sciences, is observations. That if you like what you're seeing here, it's only going to get better with more observations. And I think that's a main point that Dr. Weisburg is making. But thank you so much for your time. It's an honor that you came down to talk with us at TAC. I think we all feel way more educated about the subject.
Okay, well, thank you again for your attention and for the invitation.